Hello again, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Blaise Frederick. Dr. Blaise Frederick is an associate professor in psychiatry at Harvard Medical School and an associate biophysicist at McLean Hospital. He's also director of the Technical and Instrumental Core at the McLean Imaging Center. He received his bachelor's degree in science in physics from Yale University and PhD in biophysics from the University of California at Berkeley. His training in MR physics and his PhD thesis is entitled Three-Dimensional Nuclear Magnetic Resonance Spectroscopic Imaging of Sodium Ion using stochastic excitation and oscillating gradients. Dr. Frederick is the director of McLean Imaging Center's optomagnetic group, and his current research is focused on multimodal acquisition and processing for hemodynamic, quantitation, and physiological denoising of full data, and device development for clinical evaluation for peripheral vascular physiology. I would like to remind our audience to please address any questions at the end of the talk. For the virtual audience, please use Q&A box or raise your hand. Dr. Blaise Frederick, thank you very much for coming today, and the stage is yours.
Thank you. Hello, everybody. Yes, today I'm going to be talking about using resting state fMRI to study everything but neuronal activity. My interest is primarily in blood in the brain, not in whatever else the brain happens to do. So what can I do? Oh, there we go. Okay. Yeah, so my perspective coming at this is that the brain is basically a bag of blood, or more accurately, a series of tubes like the internet. Blood comes into the brain through the carotid arteries, goes through the arterioles, goes out to the capillaries, feeds things, goes into the venules, the veins, out through the superior sagittal sinus, and then the jugular veins. It has both a pulsatile component, the pressure wave due to the heartbeat, and the constant component of blood just sort of flowing, bulk blood flow from one place to another. And then the neurons are probably doing something with blood too, but in my perspective, they're mostly there just to hold up the blood vessels. More seriously, though, neuronal activity alters cerebral blood flow in a way that you can detect with bold fMRI, otherwise it wouldn't be something that it really does. But it's only one of many sources of variance. There's an awful lot going on in the brain and the body that can cause a bold signal, and neurons are only one of those things. Cardiac pulsations and slow variations in blood volume and oxygenation can account for up to 50% of the variance in various voxels in the brain, depending on the voxel and the imaging parameters. So, historically, what we've done with past fMRIs is we just average this away. We average forever. We do lots of different repetitions, and we hope the hemodynamic noise sort of averages away. This doesn't work so well when you're looking at resting state fMRI. Because you can't really average it away. It's the correlation between different parts of the brain that matter. And you need to be able to, if you really want to understand how this hemodynamic signal is going to affect your resting state, you need to accurately understand it, you need to characterize it, and then you can model it and remove it, and leaving the neuronal signals that you want to study. So I'll be talking about two frequency bands today, primarily, the cardiac band and the low frequency oscillation band. Which has various definitions, but we'll say 0.09 to 0.15 hertz. That's the average of the range over the literature. And then there's cardiac band that goes from 0.7 to 3 hertz when you're looking at various harmonics. And both of these are important, but the way that they're produced and the way that you understand them and the way you study them are very different. So I'm going to talk about this. The talk's going to be broken up into two different parts. So just to familiarize yourself with the spectrum of fMRI data, this is a very short TR, 400 millisecond TR brain fMRI voxel. This is obviously a pathological voxel with really loud noise, otherwise it would detect it. But this is what you expect. This is what's actually in the signal. Now, depending on your imaging parameters, you may be able to see and properly resolve various parts of the spectrum. So if you're doing 0.1, if you're doing 0.4 second TR, you can pick up the cardiac, you can pick up the fundamental of the cardiac, you can get the respiration, you can get the neuronal signal and the LFO band. But as your TR gets longer, then you're going to start to lose the ability to properly resolve some of these signals. Now, that doesn't mean that they go away. The fact that you're sampling too slowly to fully digitize them doesn't mean that they're gone. It just means that they start to get folded into other parts of the spectrum. So by the time you get to sort of classical resting state fMRI at a TR of 2 seconds, all of the junk to the right here just gets sort of folded into the part on the left where you actually care about the signal. So you would like to be able to understand some of these signals and remove them properly. So modeling is really the way to go on this if you can do it, because a spectral filter is not going to get you out of this mess. Okay, so we're going to start with LFO band noise because that's my primary input. So just a little bit of history. In 2010, back when I was doing a lot of concurrent fMRI and NIRS experiments, we were trying to understand why the signal from a frontal lobe NIRS voxel correlated so well with various parts of the brain. We made an accidental discovery. What we decided is that we were actually seeing this global signal, and that by looking at global signal in different ways from what you would do conventionally with fMRI, you could actually learn quite a bit about circulation in the brain. So here was the experiment we were doing. This was actually a bilateral motor strip experiment, alternate hand tapping, finger tapping. We got a bold signal here on the upper left-hand corner, which we could also map out our oxyhemoglobin signal and our deoxyhemoglobin signal that we recorded with NIRS and our total hemoglobin. And we saw that they agreed very well. So we had this idea that we were going to take the oxyhemoglobin signal recorded with NIRS and use that as a probe to analyze our fMRI data, because we said, okay, we're sick of applying this canonical hemodynamic response function. Let's just measure the hemodynamic response function directly, and then we'll get much better bold analysis. And that's not actually what happened. So if we look down here on the bottom, where we do our standard analysis, looking at a block time course involved with the HRF, we pick up the motor strip, very nicely. We see when you're tapping with the left hand, when you're tapping with the right hand, you get left-handed activation up in the motor cortex. Very clear and not a lot of junk. When you take the oxyhemoglobin time course recorded from NIRS, you get pretty much the same activation. You can see, the same regions are activated. You're also getting an awful lot of extra junk. And that was disturbing. And in fact, if you take the oxyhemoglobin time course from the contralateral side, which is not going to pick up your activation, you get almost as much activation all over the brain. So the fact that we get the correlation in the motor strip where we expect to see it, that's not surprising. That's what we expected going in. The fact that we see NIRS, that when we correlate with the NIRS signal, we get activation all over the brain. That was surprising because the NIRS probe can only see about 2 centimeters into the brain. It sees about 2 centimeters from the surface of the head. So there's no possible way it could be seeing the cerebellum, and yet it's correlating very well with the cerebellum. So we needed to try to figure out why that was. So one of the things, so looking at it, we found that the widespread portion of the signal was very similar in intensity in the task and the non-task regions. We also found that it's bilaterally symmetric. You can go back to the picture. See, it's very bilaterally symmetric, which is a little weird for noise. What made us think it's not noise to begin with is that bold and NIRS don't really share any imaging physics or any mechanism. They don't share any of the same mechanisms for generating artifacts. So it was very hard for us to understand what it could be that was tying these two together, unless it was something real. The real thing that's there is the head, right? They're both looking at the head. So maybe there's some physiological thing that's tying it together. At first we thought, oh, maybe we're seeing we're seeing a resting state. We're seeing two different parts of a resting state. But if you just look at the patterns, you can see it doesn't agree with any sort of resting state you're used to seeing from fMRI. So then we started to wonder if it was bold, if it was global hemodynamic fluctuations. And we know that there are prominent anti-correlated fluctuations in the low frequency band in NIRS. the oxy and deoxyhemoglobin oscillate in the low frequency oscillation band, and they tend to be anticorrelated. So as oxyhemoglobin goes up, deoxyhemoglobin goes down, because with a fixed amount of blood, the oxygen is either there or it isn't. Right? But in fact, also the volume changes. There also are low changes that are correlated between the two. And there are many known mechanisms for this sort of thing. There's Meyer waves. They're known to be low frequency oscillations seen in bold fMRI. You see blood pressure oscillations. There are all kinds of things that happen in that particular frequency band. There's also, if you look at electrogastrogram signals, which is measuring gut motility, and correlate them with the fingertip, you actually see a correlation there too. So where these things come from is not entirely clear, but it doesn't really matter for they exist and they're wandering around. So we thought that maybe we were seeing these low frequency variations in blood oxygenation and volume, that these were just passing through the brain. So the way we tested that is if these fluctuations are moving with the blood, then as we correlate the bold signal with the NIRS signal, if we change the time delay between the two, then these activations should move, right? Because then we're correlating, they should be aligned differently in different boxes at different time delays. So we thought, okay, that seems like a good idea, because we can just take our regressors and we can shift them and we can do the correlation. And that turned out to work surprisingly well. If you look here at the upper left-hand corner, you'll see that this is a map of the correlation between the NIRS regressor and the bold data as we stepped the time delay between the two from -7 seconds to +7 seconds. By the way, I can answer this later, but I'm presenting this as a way as if it sounds like we know what we're doing and we made intelligent choices and all of this. This is all just complete hack job. We were desperately trying to figure out why these things weren't aligned. We was like, oh, we're off by a TR. Oh, did I mean a TR this way? I meant a TR this way. I meant 2 TRs. So we were just flailing, and we happened to see this pattern. But what was interesting here is that the biggest signal ends up at the end of the correlation with the longest time delay, up here in the upper left-hand corner of the brain, in the sagittal slice. That's the superior sagittal sinus. That's the big draining vein down the back of your head, which you see in this nicely rotated 3D view in the lower right-hand corner. So we thought, okay, so we're just seeing blood. And we thought, well, what can we use this for? Maybe if we study it more, we can learn more about it. So we decided to do a systemic evaluation. And again, this is me presenting it as this was our bold experimental plan. What it was that reviewer one said, you're not publishing a study with one subject, you need to do more subjects. So we went back and did a whole bunch more study. We eliminated the task because originally this had been a finger tapping task. And that was a confound because we didn't want to mix up the task and the global oscillations. And we looked at 6 subjects. But the most important thing about this is that we came up with a nice acronym for it. And we've stuck with this acronym for several years, so it has some staying power. So one of the things that when I first, when I told you these were global signals, the first thing we wanted to establish before we started doing this is, are they really global? And it turns out they are. We basically took our NIRS machine and we hooked it up to every part of the body that we could stick it to. And we found the same signal in the fingertips, in all of the fingertips, both hands, toes, cheek, earlobe, tongue. We looked other places, but anyway, we saw it everywhere we looked. And what we found is that there were characteristic delays between different parts of the body, and it made sense. If you look at the correlation between the left and the right fingertip, the correlation is about 0 time delay. That makes it a little, there's a little bit of a lag. If you look between your left and your right big toe, 0 time delay. But your toe is about 3 seconds after your hands, and that makes sense because they're farther away from the heart. There's a very mild correlation with height too. So the taller you are, the farther your toes are away from your fingertips, and so the longer the delay. So we got great, we got an interesting method of measuring blood flow. So when we put this together and we ran an experiment that was actually designed for this, we actually got better activation, and it repeated the same pattern. So it came in from the center of the head, which is where the blood comes in. propagated through the parenchyma, ended up in the draining superior sagittal sinus, the transverse sinuses, and out the jugular veins. Okay, so that's great. You know, this looks cool. What can we use this for? Well, I mean, what you can do is we can use this technique to split our signal up. If we look at the low frequency oscillation band, we now have a way of parsing this into two distinct parts. We have the part that moves and the part that does not move. And which of those is signal and which is noise depends on your perspective. So the first place we came at this was from is the moving signal is the signal that we care about. Because we learned all kinds of things about blood circulation, which is really neat. By looking at cross correlation between this fingertip regressor and the various boxes in the brain, you can get sort of a relative, you generate maps proportional to RCBV. The more blood you have, the more variance in the box is going to be explained by the signal, by the fingertip signal. You can also generate arrival time maps, which is very cool. Now, I showed you those two movies before. This didn't look that interesting because normal healthy circulation is not that interesting. Blood comes in through the carotids and it leaves your head about 6 to 9 seconds later through the jugulars. It's bilaterally symmetric and it follows a very stereotyped pattern. But not everybody has normal circulation. So the first thing we wondered is what happens if we look at people with pathological circulation. The other thing that was kind of interesting about this is that we get this all for free. This all comes from a resting state fMRI experiment. We're not doing ASL. We're not doing any sort of fancy experiment. And ASL isn't even good for this kind of thing because ASL is typically limited. I mean, now there's velocity sensitivity at ASL, but at the time we were doing this, if you excited in the neck and your blood didn't get somewhere in 3 seconds, well, you're out of luck, right? Blood didn't get there as far as ASL is concerned. This doesn't really care how long the delay is because you're just looking at correlations between two noise signals. That the signal that you're looking for is not a transient magnetization that's going to decay away over a few seconds. You're looking at a pattern of oxygenation or fluctuation that's just just going on and on forever. So this is a little bit of a gear switch. This is not using an endogenous signal. I have to be fair here. This is using a carbogen challenge experiment that one of my collaborators, Martinos, was doing at Vanderbilt. But he was looking at cerebrovascular response. You'd give some carbogen, look for the response, you look out for how big the response was. And so we said, hey, what you really want to look at is when the response arrives, because that would be super cool because then you know when the blood got there. And so he let us do some analyses. And we found that it was really, Moya is really super interesting to look at using this technique. So here is an example of a patient who had severe Moya disease on the right side. So what you're looking at is, you look at the bottom here, that little scale, this is the correlation over time lag from -80 to 140 seconds. So what is arriving in the the back of the right hemisphere in this subject, something like 90 seconds after it gets to the contralateral side. So this is severe circulatory compromise. Now, Moya is bad, because with this level of circulatory delay, you're very susceptible to strokes. Blood becomes actually more, it's a non-Newtonian fluid, but opposite from the way cornstarch is. The slower it's moving, the thicker it becomes. So as blood starts to slow down more and more, you're more likely to have it clog up somewhere and give you a stroke. So you really want to deal with this. So there's a procedure for this called EDAS revascularization, which stands for some 50-letter long word, where you take an artery that's not doing something very important, and you pick it up from one part of the brain, sort of jam it down into the part of the brain that's not getting blood. That turns out to work pretty well. So this subject here is at the top, this is prior to the EDAS revascularization surgery. 6 months after surgery, you can see that the blood is arriving in the affected hemisphere very much earlier than it was before. And 22 months post-surgery, it's almost symmetric blood flow again. So this is a case where the surgery worked really well. Up until now, the surgeons here didn't have a way of assessing the quality, whether or not the surgery succeeded, other than just following the patient and seeing if they happen to get a stroke. So being able to monitor this, directly after surgery and see restoration of function is very important. And so they use this routinely now at Vanderbilt to do this sort of thing. So it was interesting, actually. You can look, if you map this onto the surface, if you map the delays on the surface, you can see that the delay times in the right hemisphere were extremely long. Again, I think they were up to 60 seconds delayed in this area. And then you can see that that function was restored over time. And you only had a small area with delayed function afterwards. And I think that was the farthest away from where they happened to jam down the artery. Although it's important to look that you can see how the circulation is all connected. Circulation didn't go back to normal; it became normalized, but it shifted around. A lot of stuff was redistributed, and when it got where, I mean, in a way that turns out to be functionally fine, but this still lets you see that the compromise hasn't been completely eliminated. It's been substantially ameliorated. Okay, so how does this affect resting state networks? Because you can imagine that this might be a problem, right? Because if you're looking for correlated blood flow, and I just told you that blood arrives with at the same, this noise signal arrives at different places at different times, and it's correlated, that's going to be a problem. So it turns out it is a problem. So we did an experiment where we took fingertip regressor and we did resting state fMRI on you have your hand up for a question? Or? Yeah. Sorry, what? I don't recall, not pronounced, but I think mostly confined to the hemisphere. Things got redistributed within the hemisphere where the surgery happened. But I wouldn't swear for the fact that it wouldn't change the other side as well. But, okay, so anyway, we did a resting state experiment. And one of the things that we had started to suspect is when you do an ICA, if you do ICA, you get a whole bunch of patterns that come out. You get a bunch of resting state networks. The thing about ICA is that ICA really hates time delay. If you have a signal and you delay it by 2 seconds, and you make a combination of these two different signals, it's going to see there's two completely different waveforms. It doesn't say, oh yeah, this is just I saw this over here that is delayed. It doesn't know that. If it sees a signal at one place, it will say this is one thing, and if it sees it somewhere else, it's going to say it's another thing. So we started to suspect that there could be some degree of this in resting state networks. And what we found is if you correlated the fingertip with the time course of the various networks that you derived from an ICA analysis, group ICA analysis, you found way greater than chance correlation with certain networks. Certain networks were very highly correlated. In fact, the red distribution here is what you would expect from chance correlation between truly unrelated waveforms. But you see on the histogram that you get, it goes way farther out than that. And in fact, some of the resting state networks have correlation coefficients up to 0.5 with the fingertip, which is not good. Now, this one in particular, the top left-hand corner, that's actually superior sagittal sinus. I mean, this is something that comes out of a lot of analyses. You do get these, oh yeah, that's a blood network. So this one you would have dismissed out of hand, but a lot of things that you actually wouldn't have called that. I mean, you have this motor strip activation, you have some auditory networks. This still has significantly higher correlations than you would expect from chance. The good news is, though, if you calculate the cross correlation and then you make a delayed copy of your fingertip regressor and you regress it out from these networks, you can remove this signal. You can, that's the modeling and removing. And if you do that, what you'll find is that the distribution of correlation coefficients becomes normalized. It goes back to what you would expect from chance, which is good. And in fact, when you look at a lot of major networks, it's interesting, you'll see that the networks don't go away. These are not, with the exception of the one that's the blood network, which was, it should go away because it's just blood. These circulatory correlations overlay what are probably true neuronal correlations. And you could probably get into some sort of deeper, some of the alignment is natural. I mean, the left and right portions of a structure are probably going to get blood at the same time because the brain is symmetric and gets blood flow. But it's also possible that different parts of the brain develop at the same time evolutionarily, and so they develop blood supplies at the same time. So it's possible that you get coincident blood flow and neuronal activation. And Molly Bright's been doing some stuff with what she calls physiological networks that seem to say about the same thing. Anyway, so if you do consider this moving component noise, which is what we were just showing, you can generate it and regress it out in a voxel-specific confound, and it removes the in-band noise. And the nice thing about this is it does this without creating the negative spurious correlations, the spurious negative correlations that you get with global signal regression. What we showed here is an experiment from 2016 where we showed that we get significantly, typically higher noise removal using our method, which we call dynamic global signal regression versus standard off-the-shelf global signal regression, static global signal regression. So what you find is that the graph on the left, you show significantly higher removal of noise in the regions with a lot of blood. But over the graphs on the right will show you that yes, you have less correlation when you when you regress out these signals. The correlation between regions is typically less, but it's greater. What's left is greater with the dynamic signal regression than it is with the static signal regression. And what's more important is you really don't create the negative correlations that you do with global signal regression. The reason for that is global signal regression is basically taking the same signal. The global signal is basically this moving signal averaged over a range of time delays. Now it's a very low frequency signal, so when you smear it over time like that, it looks kind of like itself, right? The problem is if you take a signal, if you take the correct signal with the wrong time delay and regress it out of every voxel, which is what you're doing in static regression, it's only, the time delay is correct only in a very small number of voxels. Every other voxel, regressing it out, it's going to overcompensate. It's going to subtract a copy of itself off at the wrong time delay, that's where the negative correlations come from. You don't get that when you actually apply the correct time delay beforehand. Okay, so just sort of step through over time. We've made a bunch of methodological improvements. In the process of making that paper that I just showed you, we came up with a way of recursively generating the noise regressor out of the bold data itself. So we no longer need the NIRS fingertip regressor, which is great because most of the time you don't have that. We showed that the moving regressor removes noise from a different pool than RVT and retro I-core. The retro I-core noise is basically alias cardiac, so it's a completely different thing. I mean, removing that is worthwhile, but the manifestation of low frequency band is not the same as what we're looking at here. We have checked this against DSC imaging, and we find that our delay maps come out to be about the same as what we get from DSC imaging, which is great, because if it actually is blood flowing, then it doesn't matter what the tracer is, you should be able to see it. We've also found that you can detect and track arterial blood, which it turns out you need to play a trick with that because the arterial blood will correlate negatively with most of the brain, and that's because blood oxygenation, if 100% oxygenated blood has the opposite is diamagnetic relative to tissue as opposed to paramagnetic relative to tissue. So the bold signal inverts in arterial blood. So by correlating the arterial blood, or you say if you put a voxel in the carotids and the jugulars, you get a very strong negative correlation between the two, and the time delay is how long it takes blood to go through the neck. And we've also released an open source package to do this calculation quickly and easily. It's got a nice GUI. You can go get it on GitHub, and you can be doing this kind of denoising yourself. And I strongly encourage you to. So, having come up with the bold only measurement, that actually opens up a lot of possibilities because now you can do these analyses retrospectively, right? So you can apply them to the HCP and Friends, the ABCD, OASIS dataset, ADME, UK Biobank, all of that you can run this type of analysis on. Now that's great if you want to do neuronal connectivity, you really should, because this will be removing noise that is going to be causing confounds in your analyses. But it also means that you can use these enormous data sets to study circulation in pathological populations that are well characterized, and you don't have to spend all that money needed to collect, 500 people with Alzheimer's or something. So we currently have a project to develop a cloud-based system that will apply these, will apply rapid type to these various databases. to get hemodynamic parameter estimation, but also to provide denoise data for people to then move on to other data sets. And also the fact that we can do it with bold-only data means that you can apply it in situations where even experiments you're doing now where you don't have the NIRS compatible MR device, or MR compatible NIRS device. So here's just an example. This is running rapid tide on 4,036 resting state scans from the HCP and averaging the delay. Over here on the left, we have the delay map. We can make a very detailed blood arrival map, average blood arrival map, and we can find you see the blood distribution on the right there. It works really well with default parameters in the HCP young adult. What we found is that when you move on to data sets like the ADNI and the OASIS data set and the HCP aging, people's circulation starts to get very heterogeneous as they get older. And these are people who are healthy and normal. I like to say that people develop vascular character as they age. It's not uncommon to see the same structure in the brain on opposite sides with a 5-second delay between them. The left vascular territory versus the right vascular territory. Just because I don't know, integration of all of the wine and cheese you've had in your life, build up plaque on one side, not the other. So that's likely to be very interesting going forward. And the challenge will be figuring out how to discern that from pathology, because certainly in things like Alzheimer's, you would expect much more profound deficits. By the way, I have a collaborator in Germany who is using this in the emergency room to diagnose ischemic versus hemorrhagic strokes very quickly. He can do this in 4-minute scan. He can look and say, yep, give him TPA or don't, based on delay maps. This also works very well in animals. Who's integrating? This is the analog of the 4,036 HCP scans, but this is in squirrel monkeys. And the interesting thing to look at here is, when I look at this previous map, the largest correlation coefficient on average we have is about 0.6, which means 36% of the variance in the signal is explained by the low frequency oscillation. In monkeys, it's much higher. Average correlation coefficient, something like 0.8 in a lot of the brain. So you're talking about over 60% of the variance in the brain signal is due to this just low frequency hemodynamic noise. So if you're going to be looking at connectivity in monkeys, then you probably really want to use this, because most of your signal, the vast majority of your signal is spurious. Okay, so moving along, I can get through this, I can. Going on cardiacs, can we do something similar for pulsatile signals? Can we figure out a way of pulling out the cardiac pulsation signals from the bold data itself? Spoilers, yes, otherwise I wouldn't be doing these slides. You could do it just by imaging super fast, but there's a more elegant solution, which we came up with. So cardiac signals are pervasive in fMRI, they're everywhere. They affect every time point, pretty much every voxel, but they're always undersampled, right? There's just nothing you can do about that. If you have simultaneously recorded cardiac waveform, you can use that to remove this signal using various modeling methods. But there's other things you can do as well. There's my favorite paper of 2018 showed you a really cool thing you could do with this cardiac noise. So this is Kenning Voss's method called hypersampling with analytic phase projection. What you do is you take your cardiac waveform and you apply a Hilbert transform, you get the cardiac phase at every time point. And then you think very carefully about when every time point was, when every single slice, every voxel was acquired. You take its slice timing, you take its where it is in TR. So you can assign for every single time point in every single voxel the exact part of the cardiac phase in which point it was acquired. Then you can resort that data and make a 4D projection to look at what the blood signal looks like over the cardiac cycle. And it's really cool. So here's applying that technique to a single resting state scan from the HCP. And you really visualize the pulsatile blood flow. Now, okay, the pretty picture, so what? I mean, I think it's cool. You can learn a lot about, say, the symmetry of the vasculature. And again, my collaborator in Germany uses this technique to look at pulsatile pathology in subjects with a circulatory problem. But then the question is, well, if you don't have a recorded cardiac waveform, what do you do? I mean, this relies on you knowing what the cardiac waveform is. So this is something we've been thinking about for years. We thought, well, if this signal is everywhere, in rapid time, we were able to pull a signal that was everywhere out of the data itself. Can we do that? And the answer, of course, is yes. And you can do that if you use one observation and 3 weak assumptions. So the observation is that while any given voxel is sampled only once per TR, you're sampling something at a very high rate, especially in multiband acquisitions. Typically, the scanner is reporting something at 10 or 20 hertz, right? That's more than fast enough to sample the cardiac waveform. Okay, the first weak assumption is that the cardiac signal is sufficiently coherent within a given slice that you can average all of the voxels. It's not actually true, but it's kind of sort of true, and it's true enough that you can make that assumption. It's especially true if you restrict yourself to blood vessels, because they tend to be much more distinct in time and much better synchronized. This is the one example where reviewer 2's comment actually made the paper way better. We did this originally without restricting blood vessels, and the reviewer said, if you did it with blood vessels, it would be much better. It's like, oh, yeah, it turns out you're absolutely correct. The other assumption is that the effect of the cardiac signal across slices is similar enough that you can merge them together somehow. With proper amount of filtering and normalization and all of that, you can figure out ways to merge different slices together into a time course. And then the third assumption is that the signal that you're looking at is pseudoperiodic. That will help you later. So in the example of the HCP, HCP data is 72 slices with a multiband factor of 8, which means that you have 9 distinct slices collected over the 720 millisecond TR. That means you're actually sampling something every 80 milliseconds. You got a 12.5 hertz sample rate. So the question is, can you combine that data in such a way that you can figure out the cardiac signal? So we just did it. We calculated normalized average time course for the vessel voxels in every slice and every stack of slices. We accounted for the slice time, the slice acquisition time, just by applying offsets, and then we just dropped them all in the same time course after normalization. And sure enough, that looks an awful lot like a cardiac waveform. And of course, it doesn't just look like it. We correlated with the actual cardiac waveform, which was recorded in the HCP data, the correlation coefficients were typically around 0.9 or higher. Okay, so the fine print, there's a lot of details. You can work them out. We supply a program that does all this for you. The normalization is a bit tricky. If there's too much motion, you need to censor those points, that kind of stuff. One of the things that we did is we wanted to make sure that this was applicable to any data set. So we sampled to a sort of a common time space of 25 hertz. So that you always get a 25 hertz signal out of this. And that makes possible the next thing that we talked about. Okay, so here is just an example of the top is the signal recorded not restricting to vessels. When we restrict it to vessels, it gets better. That's the actual plethysmogram. You can see the spectra look very similar between all of these. And then because it was 2019, we said, well, let's throw some deep learning on it and make it sexy. So could we figure out a way to get a more reliable, a prettier signal out of this? So the first thing we thought about was to apply a Kalman filter, because it seemed like sort of a natural thing to do. But if you have evolving parameters in a Kalman filter, it becomes a nonlinear Kalman filter, and that was too complicated for the three of us who were working on it. Just like we could never make it work. So we thought, oh, let's try some deep learning. Maybe we can clean it up with a deep learning filter. So the cool thing about the HCP is you've got high quality fMRI data, and you've got plethysmogram data. So you can generate these waveforms, and you know what they're supposed to look like. So, throw math at it, throw some GPUs at it. And we managed to come up with a CNN filter that was very good at reproducing the plethysmogram from that raw sort of noisy signal that we got. This only took about a half hour to train once we figured out what we wanted to do. We did a hyperparameter search to find the appropriate set of parameters. And so now, now we have something that if you give it fMRI data, you can get something that looks very much like an actual plethysmogram coming up. and it preserves all of the harmonics very nicely and everything. So in this example, we have, this is a similar sort of map that I showed you from the HTTP data. This is from the myconnectome data set, which is Voss imaging himself twice a week for a year. He did not record simultaneous plethysmograms, but we're able to pull them out of the data and generate very nice hyperparameter maps. So you might ask, why do this? Doesn't everybody record plethysmograms? The HTTP data, they record all of these things. The answer is not everybody does. And the other thing is that a surprising number of the HTTP data sets are bad. The plethysmogram data, there's about 12% of them are just, I don't know, thing fell off or whatever. It's just, those are complicated experiments. You're doing a lot of things. Making sure that the plethysmogram hasn't fallen off is not your highest priority. So this is a way of saving that data if you want these sorts of parameters. And just to be clear, this is not just a case of smoothing and making it look kind of nice. You actually get beat-to-beat detail of where the heartbeats occur with the plethysmogram. So you can take the plethysmogram out of HAPI, and you can feed it directly into the Physio toolbox and record, the RR interval across the entire HCP acquisition. And if you compare that to what you get from the actual plethysmogram recorded simultaneously, it's indistinguishable. So if you care about things like looking at physiological variation, like looking at heart rate variation, all of that kind of stuff, you can do that with the signal that you pull out of your fMRI data itself. And again, in the methodological improvement, we have some collaborators in Italy who have developed a method called Who Cares, following the tortured acronym that goes into the RapidType package that actually generates voxel-specific cardiac regressors to regress out, and that removes cardiac waveforms better than retroactive. You probably still have to use retroactive to get the respiratory component out, because this doesn't do that, but it's this is freely available now, and it's another way to improve your data. And so finally, I would like to acknowledge the huge number of people I've worked with over the years. Many of them do most of the work. And thank you for your attention.